I'm Peyton Bennett, this is Abigail Vorstag, and our enzyme is plasmin. What is plasmin? Plasmin is a proteolytic enzyme that acts to dissolve fibrin in blood clots. So as a serine protease, plasmin cleaves many products in addition to fibrin, such as fibronectin, thrombospodin, laminin, and von Willebrand factor. To the right, there's just the full mechanism on how plasminogen becomes plasmin, and then plasmin breaks down that fibrin, which we'll talk about a little bit later. So what do the genes tell us? So in human plasmin, it's encoded by plasminogen, which is the PLG gene. In plasminogen protein encoded by this gene is a serine protease that circulates in blood plasma as an inactive zymogen and is converted to the active protease plasmin. It is mainly produced in the liver. It's converted by several plasminogen factors, such as tissue plasminogen activator, or TPA, urokinase plasminogen activator, or UPA, kallikrein, and factor 7, which is the Hageman factor. The conversion of plasminogen to plasmin involves the cleavage of the peptide bond between arginine 561 and valine 562. Plasmin cleavage also releases the angiostatin protein, which inhibits angiogenesis. And the image to the left is the complete structure of plasminogen, as well as a sample nucleotide linkage. And then right below the image for that is the key to all of those nucleotides, and as well as their bonds. The protein itself, plasmin, is found in mammals, though plasminogen is only found in humans, wild boar, and cattle. Plasmin is a proteolytic enzyme, while plasminogen is a serine protease. Plasmin contains 7 structural domains. In addition to a C-terminal chymotrypsin-like serine protease domain, plasminogen contains an N-terminal pan-apple domain together with 5 Kringle domains. So basically, a proteolytic enzyme is a substance that causes the breakdown of proteins. A serine protease is an enzyme that cleaves peptide bonds. So the serine protease domain and the PAP domain contain important determinants for maintaining plasminogen structure, specifically in the closed form. And then the Kringle domains are responsible for binding to lysine residues present in receptors and substrates. Plasmin is activated by proteins such as alpha-2 macroglobulin and alpha-2 antiplasmin. The mechanism of plasmin inactivation involves the cleavage of an alpha-2 macroglobulin at the bait region by plasmin, which is basically just an area that is really easy to cleave. This initiates a conformational change that causes the alpha-2 macroglobulin to collapse around the plasmin, and the resulting complex has an active site that is sterically shielded, limiting the plasmin's access to protein substrates. 2 additional events that occur as a consequence of the bait region cleavage, namely, number 1 is high cysteine G-glutamyl thiol ester of the alpha-2 globulin becomes highly reactive, and 2, a major conformational change exposes a conserved COOH terminal receptor binding domain. The exposure of this receptor binding domain allows the alpha-2 macroglobulin protease complex to bind to clearance receptors and be removed from circulation. So the mechanism for plasmin breaking down fibrin, it actually breaks the cross links between fibrin molecules, which are the structural supports of the blood clot, and it actively is extremely short-lived. So fibrin degradation products are what is produced from this process, and they're just substances that remain in your bloodstream after your body dissolves a blood clot. These products are removed by the reticuloendothelial system and your other organs. Down on the bottom is an easier version of what's on the right to understand. So basically, it's showing all of the other factors that come into plasminogen converting to plasmin, and then plasmin breaking that fibrin bond.
So the paper we chose to research was called Hyperfibrinolysis Increases Blood-Brain Barrier Permeability via Plasmin and Bradykinin-Dependent Mechanism, and it was published in 2016. So hyperfibrinolysis is a condition in which the process of blood clot formation is disrupted, and there's an increased risk of bleeding in individuals who are experiencing this condition. And this condition can arise from complications due to various clinical disorders, such as like brain trauma, liver cirrhosis, and leukemia. And the researchers who published this study aimed to define plasmin's role in the induction of the hyperfibrinolysis pathway and plasmin's effect on the blood-brain barrier homeostasis. So the methods of this experiment, the researchers used mice, and the mice were hydrodynamically transfected with P. live, which is a plasmid vector system. And the plasmids they injected these mice with encoded for wild type rat PTA, which is a plasminogen activator. And plasminogen, like Peyton said, is involved in the breakdown of blood clots. And it's found in the endothelial cells that line blood vessels. And as an enzyme, it catalyzes the conversion of plasminogen to plasmin, which is responsible for blood clot breakdown. The other plasmids encoded for mutant TPA and fluorescent TPA, so they could kind of like see where these proteins were being produced. And the last plasmid encoded for zero, it was used as a control. So a large volume of a buffer containing the plasmids was injected to the mice's tails, and Evans blue dye was injected in another buffer 2 hours before the mice were killed and 48 hours after they were transfected with the plasmid. And the mice's organs were imaged using infrared fluorescence. And after transfection, the mice were treated with drugs that would either promote or inhibit plasmin generation. So their preliminary results were that hydrodynamic transfection of the wild type TPA leads to chronic plasmatic secretion of TPA. And the chronic plasmatic secretion of TPA triggered a hydrodynamic transfection of P. live TPA, which leads to the hyperophenolic state. So basically, their preliminary results just said that this system would be effective in analyzing how plasmin affects the brain, the blood-brain barrier system as a whole. So it was basically just saying that like transfection with these plasmids is effective in creating conditions that look like the hyperfibrinolic state. So these are some of their results. The hyperfibrinolysis induces blood-brain barrier leakage in a plasmin-dependent manner. So the P. live TPA expression promoted leakage, which you can see with the EB dye into the brain compared to the P. live control. So the figure B on the left is showing the P. live control, while the one on the right, the three on the right are showing that transfection with wild type TPA induces a blood-brain barrier leakage, which you can see through the fluorescent dye. And the blood-brain barrier leakage is dependent on plasmid as inhibition of plasminogen activation into plasmin, or plasmin inhibition prevented EB leakage into the brain. Plasmin also induces blood-brain homeostasis interruption through bradykinin B2 receptor disruption. And bradykinin is a peptide that promotes inflammation through capillary dilation. And the researchers' results showed that only icatibant, which is a bradykinin B2 receptor inhibitor, prevented plasmin-dependent blood-brain barrier leakage in the hyperfibrinolic mice. So this figure on the right in panel A is showing the known effects of plasmin and possible target pathways for plasmin inhibitors. Panel B is showing the experimental design. And panel C is showing the fluorescent images of TPA transfected mouse brains treated with 5 different inhibitors of the plasmin substrates as possible pathways. And essentially the only substrate inhibitor that was effective in preventing a blood-brain barrier leakage was a bradykinin B2 receptor inhibitor. So plasminogen activation triggers bradykinin production as well. So high molecular weight kininogen, or HMWK, is involved in the generation of bradykinin. And there's a lot of previous research that discusses this pathway. And HMWK is a protein that's constantly circulating in your bloodstream. And plasminogen activation triggers HMWK cleavage, which produces bradykinin. So plasmin generation mediates the production of bradykinin as well as the activation of bradykinin B2 receptors. And in this figure, the first panel is showing the Western blot of HMWK after 3 hours of incubation. And it's showing you that plasminogen activation promotes HWK cleavage. That's a result of plasmin generation. And panel C is an enzyme-linked immunosorbent assay of bradykinin levels, showing that plasmin basically generates bradykinin by HMWK cleavage, and a plasmin inhibitor prevents bradykinin production. So these researchers concluded that hypofibrinolysis induces brain barrier leakage by a plasmin-dependent generation of bradykinin and subsequent activation of bradykinin B2 receptors. And though the hyperfibrinolytic state is not completely understood, hyperfibrinolysis occurs as a result of many diseases, and the author's research provides insight into potential treatment offers that can reduce the likelihood of bleeding after injury or stroke. And here are references.